Thank you, everybody, for coming, and thanks for the opportunity of presenting. I'm John Catanzaro, and I'm CEO of Neo7Bioscience, and I'm also Chief Product Developer for Space Pharma, which are working in precision and personalized therapeutics. And it's really an exciting time to be doing this. Now, I'm going to be showing you a high-level presentation. So anyone that wants to go into a deep dive into the platform, we can always set up a time to do that. But here, I wanted to give a high-level view of what we're doing. My experience was in the clinical integrative cancer realm. I graduated from Bastyr University, and I also graduated from Bethany Divinity Seminary, which I got a PhD in medical genetics and theology, along with my medical degree. And then I've been working as a researcher since 1998. So pretty much AI prediction modeling, a lot of you may be familiar with some of the things that are involved in data mining, ranking, mapping, modeling, confidence selection. But one of the things that I want to point out is that this platform is a solution engineering platform. So in other words, a lot of the times we get predictions, but we really don't get really good, solid candidates. And the focus of this platform is on protein-peptide designs. So we actually model the protein-peptide designs by utilizing the patient's biodata. And as you can see, some of these things that some of you may recognize in your field, the platform has a robust integration, and the reason we have such an opportunity here is because my co-founder, friend, colleague, who's an executive at Elsevier and molecular biophysicist, he and I work together to integrate the technology based on live patient data. So we had the luxury of not just training sets or other data sets, but we actually have live patient data with resistant disease. And the platform was primarily modeled for cancer first and resistant cancers. And with regards to how these engineering peptides come into as the output, we first have to go through all of the stages of inputs that you can see here, looking at all of the molecular data from every angle, and then we multiplex that for complete submission into the platform. So this kind of outlines a little bit. You probably have seen some of these things too, with regards to the magnitude of mining and NLP and some of these other things, how the integrations go. As most of you are aware, Elsevier is the largest publisher in the world, and so they have access to, and we have access in our platform to all of these ability to mine in and the interrogation path. And it's really exciting because we get to see how the changes in research, we're up to date on those changes in the database. So what are we doing? We're looking at biodata, pretty much live biodata. We also access other data forms, from like the Google data pools and all these other things, right? But we're putting emphasis on mainly biodata from live patients, because we want to be able to see from patient to patient what the correlations are in the expressions of things like protein fold changing, risk marking, the expressions of both ctDNA in relation to the disease. And one of the other things here that you might see that's uniquely different in proteomics is that we're using urine proteomics. And the reason why we're doing that is because in the plasma, you can only isolate so many proteins. And so our technology that we have allows us to isolate up to 6,000 proteins. I just want to put a good plug in for Dana-Farber here, because early on in my work, we developed a team of 5 people trying to make sense out of different protein peptide expressions that correlated to the patient's own proteomic assessment. So, and we actually over the years from 2001 to 2014 had 500 patients enrolled and had the ability to look at those data points very closely. So basically, very easy input as far as, what the biodata looks like multiplexed in and what the PBIMA platform does. So basically, it's precision-based immunomolecular augmentation, because what we're doing is we're looking how to augment the pathways, so that pathways specific in the disease expression. So if we look at protein-peptide interactions and all of the other things that come with it, we have to first know what the patient's overall disease expression is. And the data, gives us a very good look, not just as a baseline, but also interval changes with any kind of intervention. So the discovery basically was something kind of dinosaurish. What we did is we isolated proteins from the urine, and then we early on, back in 1998 in resistant cancers, gave those proteins after they've been column filtrated and isolated the peptide fragments and gave them back to the patient under a clinical IRB. And amazingly, we had patients that were beginning to demonstrate tumor regression and significant change in their disease expression. Then what we did is in generation 1, we moved into adding the more sophisticated proteomic identification and HLA phenotyping. Back then, as you know, was pretty much in its basic form. And then we did manual sequence selections that actually worked for the patients quite well. And then generation 2, I retired from medicine and I came back in 2019 and formed the company with my co-founder. And we actually integrated the whole technology platform to include all of the elements that you see there with an auto sequence development. We didn't have checkpoints in generation 2. We added checkpoints to generation 3. So now what we're able to do is we're able to select out peptide-associated checkpoints instead of antibody checkpoints. And in the design, we can actually match these checkpoints to the primary targets so that they would have a bispecific or a trispecific action in the patient. So one of the unique features of the platform is that we're looking at the hallmark expressions, and probably most of you have seen Weinberg's early work in hallmarks, and now, of course, we've got about 15 hallmarks that we know of, and the PBIMA platform actually has these 15 hallmark expressions as part of the ranking abilities, one of the algorithms that are part of that. And as you know, cancer is a multi-disease within a disease. Now, this is a KEGG pathway. It's old. But I wanted to just put this up because it demonstrates the complexity of the pathways. And understanding what the real patient data expression is so essential, because there's no possible way you can have a mitigating effect if you don't know what pathway you're going to be focusing in on. And if most people do know that in drug discovery, because antibodies and checkpoints have to be pretty much designed to be focusing in on specific pathway interface. This is just a little bit more of some of the targets that we go after. Another slide to show that from the standpoint of looking at angiogenesis, and we go after these targets as well. And then we go after mitochondrial signal targets as well in the design. So we look at, your various HSP10, 60s, some of the things that you see here, ATF5, NFR1, all of these different genetic nuclear genetic expressions, and we can also look for mitochondrial expressions in the mitochondrial genome, but a lot of the times the mitochondrial genome doesn't give us very much solid data, right? A little bit more on, this is really interesting because we're talking about, again, mitochondrial influences, and then the relationships of lipid metabolism in resistance in various type of cancer treatments. So in our platform, we're modeling to where we can actually, if we do see these signal pathways and fold expressions where they're definitely affected in a negative fashion, we can mitigate in those pathways. Now, most of you this is a little bit more of the lipid synthesis transport modeling. Now, I just wanted to say that when we're looking at different signal pathways, we're not just going after mutations. There are a lot of protein expressions that are out there that are disease-related expressions that are not associated with mutations. So they're called epigenetic alterations. So epigenetic alterations are specific when you want to try to type to see what kind of cancer resistance you have going on, and the patient's data is pretty good at giving us some of those clues. So as you can see here, we're familiar with germline mutations, somatic mutations, but I really want to bring out the fact that there are epigenetic alterations that are specifically protein expressed in the urine that we can pick up really abundantly. So a little bit of the epigenetic effects on protein expression. Most of us in the room probably understand. A little bit on the methylation movement, hypermethylation. This actually comes into play also in longevity and aging as well. We really focus in on these type of alterations, and especially when we're seeing a lot of resistant disease and metastatic activity. Just a little bit more on non-coding RNA and chromatin remodeling, two very important parts in regulating tumor suppressor genes. And then, of course, some of the environmental aspects that can affect it, and some of the reversibility of epigenetic changes, which we've actually seen reversibility of epigenetic alterations related to the disease, resistant disease. Not just in cancer, we've also seen it in other disease types that we're looking at, neurodegenerative disease as well. Now, checkpoint targets, there's a lot of them that are being developed, and probably a lot of antibody development here to these specific targets. And we're all familiar with the PD-1, anti-PD-1, and CTLA-4, LAG-3 is kind of like a new kid on the block coming up. TIM-3, CD27, all of these here in our PepCheckX pipeline, we have the ability to model every single checkpoint from a standpoint of peptide binding affinity to the pocket. So it's really important because we've actually studied antibodies to the binding pocket, and we've seen that peptide structures have some better binding affinities than the actual antibodies themselves. So maybe if the antibodies are pieced out, where segment antibodies or you got nanobodies or things like that might have a little bit of a different turnout, but this is an important fact because there's a great paper out there right now that talks about the myriad of different peptides in PD-1 that have better binding affinities. So advancing checkpoint targets, I think, is something in the peptide realm that needs to really get, really needs to explode and take a good position in the environment, because the druggable targets, we can actually get a lot more effects that are very positive, because as you know, in antibody developments, when you're going specific to a target, you can have a higher preponderance to off-target reactions, resistance over time, and with a good designed peptide to a specific target, you're going to have less of that happening. And of course, that needs to be studied. There's still some theoretical things surrounding that, but what we're seeing in silico is pretty good, reliable data that shows us that that peptide development, especially for engineering, is got some significant promise. So benefits of carefully trained AI. Now, I wanted to mention that this AI platform is not just artificial intelligence. It's combined with human intelligence. So we're not just letting this thing fly and it does all of its prediction without having a continuous training input. So our science and data team are always curating and doing training inputs, and we're constantly making modifications to the platform. And I wanted to bring that out because the AI HI integration is where we need to really focus, not just turn it loose for just in silico management and AI integration alone, because we can't underestimate the power of the scientists out there, the data experts out there, the people that are working hard in the field. All of these inputs that happen, this amazing amount of changes that happen on a daily basis in research. And we want to capture all of that, and these scientists that are working hard to develop that. So our integration, we're very careful to be sure that that blend is present, and that the people that work with us know that. So the benefits, these are just outlining the benefits where, we're using the functional exome as opposed to using the genome by itself, right? So the genome has a lot of noise, and you have to filter a lot of noise out before you get to your real solid element. So we're using the functional expression genome, the full RNA transcriptome of both the normal environment, the tumor environment, and we're looking at ctDNA integration as well. And we look at all of the correlation of all that data set coming in through the platform. And then the layering of the urine proteomics, which is putting the specific proteins in there that are highly specific to the disease diagnosis itself. And then another important feature, of course, is the HLA phenotyping that we get through the functional exome and the RNAs specifically. And then we get that expression because this is unique to the patient. It's unique to the patient's compatibility and susceptibility in the disease process. So we get to know a lot about what is a better match and affinity for the patient by matching the immunophenotyping mapping. And then just a little bit more here. I won't go into all of these, but the slide said kind of this nice article that kind of demonstrates variant relationships and some of the benefits of some of the disease scoring that we actually have in our algorithmic engineering. Then we have the total admit process integrated in, where we're looking at modeling for that to demonstrate the best molecule for lowest toxicity, better PK value, looking at PD studies. And then also we are looking at for manufacturability. So like in other words, does it have the right polarity? Does it have the right hydrophobicity? Does it have the right characteristics to be structured with a small nanocarrier, right? And our nanocarrying right now is we're looking at, we actually have in process the movement toward doing scaffolding with Janus. And then we also have our designs in what we call a PEG design, small PEG with an aminohexanoic acid bridge, which allows us to couple different molecules together. So on individualized therapeutics, we're moving from a one-size-fits-all model, and of course the precision model, as everybody's aware of, and precision targets, because that's the focus here in research, is getting better precision targets. We're moving to the individualized therapeutics. So the movement and shift in the paradigm is to get out of just a general one-size-fits-all, because it's a good it's good for pharma, but it's not necessarily good for the person. So, and I just feel like I need to say that because when we get into individualized therapeutics, we see people making some significant progress in beating diseases that they wouldn't have any other options to beat. So when we're looking at protein peptide, the reason why I say protein peptide is because the cell-to-cell signaling and the protein-to-protein interactive pathways are very robust, and we've got to know how the communication happens between them. We're in development in a lot of these things here and getting better all the time because we're getting more data coming in, live data. And as you can see, when we're using short chain peptides, they're much more effective than if you were to take a long chain and try to, then you actually run the risk of more antibody-dependent issues, which you don't want. So the process basically for the patient is pretty straightforward. It's about a 7 to 8 week process from time of submission of all the samples for the multiplexing of the data to run it through the augmentation platform and then develop the output design. And the output design for cancer is 10 primary targets with 3 matched checkpoints that are matched to the patient's disease pathways. So they're mitigating peptides, and they're pooled, lyophilized, and administered to the patient under a clinical trial IRB setting. So compliance and regulatory, we're going to be seeing some upcoming things in development. Right now, a lot of physicians are doing physician-initiated INDs for patients that have no other options. And of course, the FDA's doing a nice job in accommodating these type of submissions. The other route is the right to try, and then there's what they call the right to try 2.0, which allows for a patient to under 2.0, which is not in every state. 2.0 allows the patient to choose a treatment that is not approved by the FDA and is not even considered an investigational product of a pharmacy of pharma. So they want to do something that's unique, and so they want something innovative. They're going to go under the right to try and right to try 2.0. And then, of course, the right to prescribe, which is a physician right movement for unapproved investigational treatments, individualized treatments. Now, this applies to individualized treatments because the FDA knows that this is coming down the pike, right? More and more patients are calling for it, and there are a lot of physicians that are out-of-the-box thinkers that are saying, we want this to be something. We want to make an impact on patient disease. We want to defeat, resistance, and this is kind of where we have as a general guideline for compliance and regulatory at this point. It's very basic. And then references, if you get a hold of the slide, you can actually, the references are all listed in the notes. You can click on hyperlinks. So that's the other thing I wanted to mention, the space pharma work that we're doing. We've actually completed 8 missions on the International Space Station. And we're studying the current mission, which is the 9th mission, is studying 2 cancerous precision drugs and 1 Zeparan, which is a prion protein drug for rare disease. And that mission is going right now. That's the 9th mission. And I actually am working along with Space Pharma for the last 2 years in the product development movement. So I have the benefit of being able to utilize what Neo7Bioscience has accumulated in the technology and layering it into the prediction systems that we're implementing in microgravity. So it's pretty exciting. That's all I have. Thank you.
Thank you, John. Any questions for John? While you're thinking about them, I have quite a few. First of all, why study medicine in space? What is it about zero gravity that there's a rationale?
Yes, the microgravity. So if you look at peptide, if you look at protein intermediates now, that's kind of a big focal point where you get loose structures in the proteins, and you're able to have a better malleable engineering to fit a specific molecule where you want it, where you wouldn't be having that ability in a gravity environment. So the microgravity allows the engineering to happen to where there is a looser fold, and that looser fold allows for a better discovery molecule, whether it's a small molecule or an antibody or even protein peptide. It doesn't matter. It's that's how it's done up in space. Yeah.
Interesting. Yeah.
Yes.
You just use.
You got to use the mic.
Okay, sorry. So in the beginning, you had mentioned about Elsevier, publication, a lot of data you have used. Any patient consent issue that, you have a lot of data from past and using it for future application?
Yeah, it's a very good question. We actually obtained permission from every one of those patients. So, yeah.
Great. And second,
Yeah.
I thought proteinuria is like a normal condition. We normally don't excrete a lot of protein. So are these kidney problem?
Well, that's a good question too. So in our technology, we're doing direct, proteomics, and we're doing exosomal proteomics. So we're able to do both and see what the correlation is between the two. So now when you're looking at proteomics, it's a little bit different than proteinuria in the urine. It's because you have normal criteria and then you have disease-related criteria. We've had the ability over those 13 years to model what we know is not normal, right? So we have that normal comparison to what we see related to the patient's disease expression that we capture in the proteomics. So now urine proteomics is much better because plasma proteomics, you're not going to be able to capture as many of those proteins because they're bound. Most of them are bound in the plasma. So you actually have to go to the urine. And so that's a very big field of study right now. Urine proteomics is a very powerful tool. Very powerful.
So, we express about 20, 30,000 genes regularly. So that's a lot of protein. Do you get those in urine? I mean,
yes.
Most of the housekeeping.
Yes, and the interesting thing, that's another good question, because we've had early on where, we're looking at the total, remember, the whole biodata picture, right? So we see something that there shows no mutation associated with particular gene path, gene protein transcription path, 0 mutations, but yet the patient expresses it out in the urine. So it becomes a very hot biomarker.
Yeah, interesting. Thank you.
Just to continue on the urine question. So is the dogma true that if you're bigger than 50 kilodaltons, you don't get filtered? Or do you see big things in urine?
You can see big things in urine. So what happens when you're doing the direct volume and the direct proteomics and the exosome proteomics is you get a better picture for comparison. So your PSM values that come out, right, are correlated to demonstrate that, does the data check against itself, right? Does it verify? And yes, you can get larger molecules there. Like, for instance, albumin is a very large protein. But we all know that albumin, we're not looking at like the largest protein in the bunch. We're looking at something that's related specifically to the cancer pathway.
So was a question at the back? Okay. As we have just a few more minutes, can you say a little bit more about the kinds of peptides you find and why do patients have functional peptides?
Yes. Okay. So now peptides are specific to transcription events. So when we're seeing a problem with any type of transcribed movement, so your DNA is kind of like the commander of the ship, and then you move into the RNA transcription translation movement, and then you start seeing specific relationships to those protein gene pathways, right? So now remember, this integration is kind of like zooming in on a nano level to see exactly where that specific issue is, where the highest immunogenetic break is, and how we can match the peptide to that specific part of the pathway. So now we can have 2 patients that have the same diagnosis and they have the same gene protein, but they have 2 different sequences, right? Yep. That are modeled to the pathway. So I hope that answers the question.
It begins to.
Yeah.
Any other questions? The only I think I'd never heard of the patient access right to try.
Yes.
So that's an interesting paradigm.
It is. And we're going to start seeing more of that because patients are really more proactive in their wanting to pursue individualized therapies. Yeah. So, yeah, we'll see more of that happening.
Great.
Yeah.
Okay. Thank you very much, John.
Thank you.